Let's dive into a key concept in blood disorders. We're going to explore a process that's absolutely vital to understanding how our blood functions and what happens when things go wrong. This is a cornerstone of hematology, so pay close attention. Hemolysis and red blood cell fragility. We'll define what these terms mean and why they're so important in diagnosing and understanding various medical conditions. Red blood cells are like delivery trucks, the workhorses of our circulatory system. They are constantly carrying oxygen from the lungs to tissues throughout the entire body, ensuring that every cell receives the vital oxygen it needs to function. They must be flexible and durable. These cells are not just simple containers, they are highly specialized structures to squeeze through tiny capillaries without breaking. These capillaries are so narrow that red blood cells often have to deform to pass through, highlighting the importance of their flexibility. Hemolysis is the premature destruction of red blood cells, a process that can have significant consequences for the body, releasing hemoglobin, the oxygen-carrying protein, into the plasma. This release can alter the appearance of the plasma and indicate underlying issues. Normally, old or damaged cells are cleared by the spleen and liver, which act as the body's natural filters, removing these cells in a controlled manner. But excessive hemolysis signals pathology, indicating that something is disrupting the normal balance of red blood cell production and destruction. Fragility refers to how easily red blood cells break under stress, a key indicator of their overall health and ability to withstand the rigors of circulation. Healthy cells are pliable, able to bend and deform without rupturing, but fragile ones burst easily, leading to hemolytic conditions, where the rate of red blood cell destruction exceeds the body's ability to produce new ones. Testing for fragility helps diagnose various diseases, providing valuable insights into the underlying causes of red blood cell disorders. From inherited anemias, genetic conditions that affect the structure and function of red blood cells, to autoimmune disorders, where the body's own immune system mistakenly attacks and destroys red blood cells. Two main stresses cause hemolysis. These stresses can be broadly categorized based on their nature. Osmotic, water balance, referring to the concentration of solutes in the surrounding fluid, and mechanical, physical force, involving physical trauma or stress on the red blood cells. Osmotic fragility and mechanical fragility are tested using specific laboratory procedures. Each reveal different problems with red blood cells, providing a more complete picture of the underlying issue. Understanding why and how these cells break is crucial for effective diagnosis and treatment, is foundational for diagnosing blood disorders. It allows us to pinpoint the specific mechanisms at play. By mastering these concepts, you'll be prepared to recognize and interpret hemolysis in clinical practice, and to approach complex cases with confidence. This is your starting point in hematology, a journey into the fascinating world of blood and its disorders. To understand why red blood cells burst, let's revisit osmosis. Osmosis is water moving across a cell membrane from low to high solute concentration. Red blood cells maintain a delicate balance with plasma, which is isotonic at 0.9% salt. In a hypotonic solution, water rushes into the cell, endosmosis, causing it to swell. The cell can expand up to 70% before bursting, thanks to its biconcave shape. If water keeps entering, the membrane eventually ruptures, spilling hemoglobin. This is osmotic hemolysis, a key reason why IV fluid composition matters in medicine. Understanding this process underpins diagnostic tests for red blood cell health. We'll explore these tests in detail next. Osmotic fragility measures, in essence, how easily red blood cells hemolyze in hypotonic solutions. It's a test that reveals the resilience of red blood cells when faced with varying salt concentrations. This test is particularly useful because it highlights the structural integrity of the red blood cell membrane. Healthy cells have extramembrane, allowing them to swell before bursting. This extramembrane acts as a buffer, providing flexibility when the cell is exposed to a hypotonic environment where water rushes in. In hereditary spherocytosis, cells are already spherical, lacking this reserve. They've lost the biconcave shape that provides the extra surface area, making them vulnerable. Spherocytes hemolyze quickly in mildly hypotonic solutions, a diagnostic clue. This rapid hemolysis is a key indicator of the disease. Conversely, cells in thalassemia or iron deficiency anemia are flatter with more surface area. This increased surface area is due to an increased surface-to-volume ratio. These target cells can absorb more water before rupturing, showing decreased fragility. They can withstand lower salt concentrations for a longer period. The relationship between cell shape, surface area, and fragility is crucial for interpreting lab results. Understanding this interplay allows for a more accurate diagnosis. Recognizing these patterns helps narrow your differential diagnosis. It allows you to focus on the most likely causes of the patient's condition. Osmotic fragility testing is a powerful tool for identifying red blood cell disorders. It provides valuable insights into the health and function of red blood cells. Always consider both increased and decreased fragility in your workup. Each provides unique information about the underlying pathology. This understanding guides your approach to anemia and related conditions. It helps you select the appropriate diagnostic tests and treatment strategies. Mastering this concept is essential for clinical practice. It's a fundamental skill for any healthcare professional dealing with hematological disorders. Now let's talk about mechanical fragility, how red blood cells handle physical trauma. The circulatory system is turbulent and cells are constantly squeezed and stretched. Healthy cells withstand this, but defects or harsh environments increase fragility. Artificial heart valves or damaged vessels can shred cells, causing mechanical hemolysis. This produces schistocytes, fragmented cells seen on blood smears. Schistocytes signal microangiopathic hemolytic anemias like TTP, HUS, and DIC. Fibrin strands in vessels act like nets, slicing red blood cells as they pass. Recognizing mechanical fragility helps identify life-threatening conditions. Hemolysis isn't just about cell defects. The journey through the bloodstream matters too. Always assess both intrinsic and extrinsic factors in hemolytic anemia. The Osmotic Fragility Test exposes red blood cells to saline solutions of decreasing concentration. Prepare a series of tubes with saline ranging from 0.9% to 0%. Add patient and control blood samples to each tube. Incubate, then centrifuge to separate intact cells from hemolyzed ones. Measure hemoglobin in the supernatant to quantify hemolysis. Plot results. Hemolysis versus saline concentration creates the fragility curve. A left-shifted curve means increased fragility, as in spherocytosis. A right shift indicates decreased fragility. This test links cell structure to clinical diagnosis. It's a fundamental skill for interpreting anemia workups. Mechanical fragility testing simulates physical trauma to red blood cells. Blood is agitated with glass beads, mimicking turbulent blood flow. After agitation, compare hemolysis in the test sample to a control. Measure free hemoglobin to quantify mechanical fragility. High fragility suggests membrane defects or vulnerability to physical stress. This test is mainly used in research but highlights the importance of cell resilience. Understanding both osmotic and mechanical fragility is key for comprehensive diagnosis. When does hemolysis occur in patients? Causes are intrinsic, cell defects, or extrinsic, external destruction. Intrinsic causes include hereditary spherocytosis, G6PD deficiency, and sickle cell anemia. G6PD deficiency makes cells vulnerable to oxidative stress from drugs or infections. Sickle cell disease causes rigid sickled cells that break easily. Extrinsic causes include autoimmune hemolytic anemia, where antibodies target red blood cells. Mechanical causes like faulty heart valves or microangiopathic anemias physically destroy cells. Infections like malaria, drugs, toxins, and severe burns can also trigger hemolysis. Malaria parasites multiply inside red blood cells, rupturing them. Always consider a broad differential for hemolysis. Identifying the cause directs your diagnostic and treatment approach. Recognizing patterns is vital for patient care. Hemolysins are substances, often proteins or lipids, that cause hemolysis. Many bacteria, plants, and animals use hemolysins as weapons. Two main types, pore-forming toxins, like streptolysin from Streptococcus, and enzymatic toxins, like alpha-toxin from Clostridium perfringens. Pore formers punch holes in cell membranes, causing rapid lysis. Enzymatic toxins degrade membrane phospholipids, destabilizing the cell. On blood agar, hemolysins create visible patterns: beta hemolysis, complete clearing; alpha hemolysis, partial greenish; and gamma hemolysis, none. These patterns help identify bacteria in the lab. Understanding hemolysins links microbiology to clinical diagnosis. Recognize their role in infectious diseases and lab interpretation. Let's take a moment to recap the key concepts we've covered regarding hemolysis and red blood cell fragility. This is crucial knowledge that will serve as a cornerstone as you advance in your medical career. Remember, hemolysis, the premature destruction of red blood cells, and red blood cell fragility, which is their susceptibility to damage, are absolutely central to the field of hematology. These factors play a critical role in understanding various hematological disorders. Red blood cells are constantly subjected to a variety of stresses within the body. They must be able to withstand both osmotic stress, which relates to the balance of fluids, and mechanical stress, which involves physical forces. When red blood cells are unable to withstand these pressures, failure occurs, leading to their breakdown and ultimately resulting in hemolytic anemia. This is a condition characterized by a shortage of red blood cells due to their premature destruction. By now, you should have a solid understanding of how osmotic and mechanical fragility tests are performed in the laboratory. You also understand the principles behind these tests, and how they help us assess the integrity of red blood cells. More importantly, you should now know what the results of these tests actually mean in a clinical context. You should be able to interpret the data and understand how it relates to the patient's condition. Recognizing abnormal red blood cell shapes such as spherocytes, which are small spherical cells, is also crucial, and being able to identify schistocytes, which are fragmented red blood cells, is equally important. Recognizing these abnormal shapes, along with understanding different hemolysis patterns, will ultimately guide your diagnostic approach and help you determine the underlying cause of the patient's condition. Remember that these concepts are not just abstract ideas confined to textbooks. They are practical tools that you will use every day in real patient care scenarios. Utilize fragility tests strategically to connect the dots between laboratory results and the underlying disease processes affecting your patients. Always think critically and consider the various potential causes of hemolysis, including inherited conditions, immune-mediated mechanisms, mechanical trauma to the red blood cells, or even infectious agents that might be involved. Approach the process of ordering and interpreting tests with the mindset of a clinician, not just a laboratory technician. Think about the clinical implications of each test and how it will contribute to your overall understanding of the patient's condition. This foundational knowledge that you've gained is your starting point for developing strong clinical reasoning skills. So keep building upon this foundation, cultivate your curiosity, and always remember to ask why to deepen your understanding. Thanks for watching. We hope you found this helpful. And please subscribe to Joen Academy for more educational medical content.